Today we'll hear from Dr. Conway. Dr. Conway is currently a professor of biomedical engineering at The Ohio State University. Dr. Conway received his PhD from Georgia Institute of Technology and Emory University in biomedical engineering in 2009. After his PhD, Dr. Conway completed his postdoctoral training at the University of Virginia at the Cardiovascular Research Center. Dr. Conway is a very accomplished researcher. He has over 45 publications between his own first authors as well as publications from his other students and collaborators. He has given over 35 invited seminar presentations, and you can add today as well, so 36. Aside from his publications and talks, he's overall just a really active mentor. He has served as a mentor for 5 postdocs, and he has one currently. He has had multiple master's and PhD students and over 30 undergraduates. I find that crazy. Kudos to you. The students he mentors are also extremely successful during their time with Dr. Conway as well as afterwards. He has had 7 students, past students and current getting NSF awards either during his training or shortly after. And he even has had 1 student receive an F31 after his mentorship during her PhD in Vanderbilt. And also, Dr. Conway is one of the first PIs that I reached out when I first started my PhD, and he's always been extremely kind and very helpful. So I'm excited to hear his presentation on the mechanobiology of lamins, vimentins, and keratin in intermediate filaments. So with that, I'll stop sharing my screen and you can start sharing your slides.
Sure. All right. So thank you, Clara, for such the kind introduction. And it's really my pleasure, right, to be able to talk to this group. I have loosely followed this seminar series for quite a while, and I've had some teaching obligations that have kept me away certain semesters. I'm really excited and honored to be able to present to you all some of the data, and a good bit of today's data will actually be unpublished. And so as a consequence, some of it may not be fully well understood, but I thought it'd be great to share it with this community and potentially get some feedback as well. And what I really want to talk today is really about our efforts to try to understand intermediate filaments and specific ones like lamins, vimentin, and keratin networks. And then I'll spend the second part of the talk talking a little bit about some work we're doing with desmosomes as well. So my group overall is kind of, sometimes I feel like a little bit dabbling in too many areas, right? And sometimes that's driven by my curiosity and sometimes by my students' curiosity. But if I have to sort of summarize this in sort of a 30-second overview, I tell people half of my group is really focused about developing tools for understanding cell biology. We're very interested in mechanical forces. And if you caught Karsten's talk with this group a couple of weeks ago, he really introduced this idea of these fret force biosensors. It's no coincidence we're doing similar things because we both trained together in the same postdoctoral lab. And I view this as sort of a contribution because I'm an engineer, right? And I like to build tools and understand things like that. But I think it's important not just to build the tools, but also to use the tools to understand and show people how these forces could be important, because it's of no value to show you that a certain protein experiences force if it can't help you understand why those forces are important. So today's talk will be centered around 2 things. The first half will be about actually using these biosensors to study forces on nuclear lamins, vimentin, and a little bit of a sort of a precursor to where we're trying to go in the direction of keratins. And then the second part, I really want to spend some time talking about our efforts to try to understand why are desmosomes important in epithelial tissues? And by epithelial tissues, I really want to understand what they're doing outside of the skin. All right, so for the first part, I'm going to start with just kind of an overview about how I think and how, just a general idea of mechanobiology. This is a really old figure that came from a 1995 Peter Davies paper, where he was sort of going over how endothelial cells, which are the cells that line blood vessels, how do they adapt to the forces of fluid shear stress, right? But easily this cartoon could be sort of adapted to any kind of force like stretch or pressure forces as well. And the reason I really like this cartoon is because, again, it was from the 90s, before we really had a good understanding that the cytoskeleton is so integrally integrated into the nucleus, right? And what he sort of postulates in this review article, right, is that even though you may have forces that are applied to the outside of the cell, these forces can be rapidly transmitted to regions that might experience large stresses because those regions are fixed, right? And if we sort of blow up these regions, right, we think about focal adhesions, we think about cell-cell adhesions, or if we think about the nuclear link complex, oftentimes these are very busy structures, right? And so these are images I've just poached from Google Images, right? Just to highlight that as someone who's not a cell biologist and someone who has trouble remembering and memorizing things, it can be very daunting when you look at a focal adhesion to try to understand which molecules are important in a structural sense and which ones may be important more in a signaling sense. So there's a real need for tools that can have protein specificity to help us understand what's going on mechanically in these structures. So Karsten already introduced this. So if you didn't catch this talk a couple weeks ago, I encourage you to go back on YouTube and rewatch it. It was really an excellent talk. Basically, the idea in these fret force biosensors is we're relying on this technique called FRET. And so this is a technique where you get energy transfer. So a donor ends up exciting an acceptor. And anytime this happens, it happens only when things are really close together. And the closer they get together on a nanometer scale, the more energy transfer there is. And so the trick that Karsten was the one who actually developed this along with Brent Hoffman in Martin Schwartz's lab is to sort of find the right elastic peptide that would connect these two, the acceptor and donor, the fret pair together. And this allows you to have a sort of fret force module that would, or you can think of it as a strain sensor that has an inverse relationship. So as the tension is increased, the distance increases, and that causes a reduction in force, right? And you can see some of their data in their paper where they had collaborated with optical tweezers pull on this sensor, and you can see a corresponding drop in fret. And this particular peptide that we're using comes from spider silk protein called flagelliform, and its dynamic range is of about 1 to 5 piconewtons. And this is a really critical feature of these fret force biosensors, is having the right sort of dynamic range, because you don't want something that's too stiff or too compliant. And Karsten has really led the field in this biosensor area by coming up with new elastic peptides that could sort of extend this dynamic range. And this is just sort of an overview slide of some of my work, but also the work of others, including Karsten and other groups who have tried to use this strain gauge, inserting it in the middle of a protein, and trying to understand in places like cell-cell adhesions, where you have tight junctions, adherence junctions, and desmosomes, that all of these proteins can experience mechanical force, but oftentimes they're loaded in different ways, whether that be temporally or different magnitudes of force. My group has really also led this effort in applying this technology to the nucleus. We published a force sensor for Nesbrin-2. We have a recent sensor for lamin A that I'll go over today. And then we have some work that I won't show today, but if people are interested, I'd be happy to chat offline about looking at forces on the nuclear pore complex. And then what's not pictured is focal adhesions, where Karsten and other groups have really led that effort. And I'll talk today a little bit about some of our efforts to think about ways to also apply this technology to looking at cytoskeletal structures, and especially with regards to intermediate filaments. So my interest in intermediate filaments actually started with my postdoctoral work. When I joined Martin Schwartz's group, Karsten and Brent had already developed this fret force biosensor module. And it really gave me the opportunity to be able to apply it to a system that I was familiar with my PhD work, which was how endothelial cells adapt to fluid shear stress. And Martin had previously published that V-cadherin and PCAM, along with VEGF receptor 2, form a complex that's really absolutely essential for endothelial cells to sense and respond to fluid shear stress. And he knew that from that previous work that these molecules are active within a matter of seconds of flow. So the prevailing idea that we had at the time is that one of these molecules would experience an increase in force, right? Because we're going from no flow across the endothelial cells to suddenly an onset of fluid shear stress on those cells. And what was really surprising to us, actually, and so I think this is sort of the value of making these sort of careful measurements, is oftentimes your intuition can be incredibly wrong. So when you apply fluid shear stress to endothelial cells, it turned out that V-cadherin actually relaxed. Which caught us totally off guard, made us really rethink, are these sensors actually working correctly? But it turns out we had other ways of measuring forces on focal adhesions and cell junctions, sort of more global forces, and the cells actually themselves relaxed at the onset of shear stress. And if you go through the math, fluid shear stress is actually a really small amount of force compared to the forces that the cells are just naturally exerting when they're attached to a surface. So in hindsight, right, we shouldn't, our intuition was sort of off on that. But we developed a new sensor as well for PCAM1, and I was able to show that that force actually increased at the onset of shear stress. And it really depended on vimentin. And so that was something that came up on a mass spec screen. And then I was able to go in and deplete vimentin with siRNA or knock it down. And that also inhibited the adaptation of endothelial cells to shear stress. And other groups have shown this, namely Peter Davies had some really elegant work where he showed that vimentin networks deform at the onset of shear stress, right? It could be important in mechanosensation of flow. And this, when I started my own group, this really led me to ask the question, what are the mechanical roles of intermediate filaments? I mean, they're really interesting structures. I mean, they depend, varying on the cell type, you have different intermediate filament proteins. And so people in the field have postulated, this could be a way that you could mechanically tune cells, right? Muscle versus epithelial cells versus neurons. And so as engineers, we oftentimes make simple models, right, that are probably too simplistic. And so Don Ingber has sort of coined this term cellular tensegrity, where he, in the 90s, put out this idea that actin and intermediate filaments would be best suited to bear tensile forces, and things like microtubules would be better suited to bear compressive loads, right? And he also was able to sort of demonstrate or really nicely show that cells, just by existing, right, and being attached on a surface, already are experiencing large forces, right? So oftentimes you don't have to poke and prod the cells. There are structures already in the cell that have really large forces. And if you teach cell biology like I do, oftentimes I grab images from the molecular biology of the cell textbook, and you can think about this also in an engineering perspective. That actin is quite good under tensile forces, but microtubules are not really that great. But the beauty of intermediate filaments, right, is oftentimes they are the strongest material in the cell. And then you can just look at structures. So in this case, this is keratin. And you can see, just like a piece of rope, right, the keratin is taut at cell-cell adhesions, right? And that also would suggest that intermediate filaments experience forces, right? And I remember having a discussion with Andrew Kowalczyk right at the time I was starting my group. And I said, are intermediate filaments under load? And he said, yeah, it's obvious, and we talked about pictures like this. And I said, well, where's the data? And he said, well, that's your job. And so I just remember that moment of like, oh, so I can just follow up on something that people hadn't really thought about. So one of the things where we got interested in intermediate filaments, we had a little bit of trouble, though, thinking about ways to develop and design biosensors that could measure forces on those structures. So the traditional way that, and Carsten probably already mentioned this as well during his talk, is that we take this sort of molecular strain gauge, which is essentially 2 GFPs. So it's a quite bulky structure, and we insert it in the middle of a protein. And if you know anything about tagging your proteins with GFP, right, typically you would put it at the N-terminus or C-terminus, right? It would be kind of strange. I mean, there are a few proteins where people have found internal sites, but it's sort of unorthodox, right, to put it in the middle of the protein. But we have to do that because that's how we're going to measure tension. So we oftentimes spend a lot of time trying different sites and a lot of control experiments to really prove to people that we really haven't messed up this protein, right? Because now we have a much bulkier protein. It may not behave correctly. So when we started to think about things like nuclear lamins, I thought this approach isn't going to work, right? Because essentially, if you think about the nuclear lamins or the cytoskeleton, like an engineer like me, I think about them like Legos. And if you have a different size Lego, right, it's not going to stack up correctly. So these cytoskeletal networks probably wouldn't polymerize very well if you had different size molecules. So we came up with an alternate idea, which was to take our strain sensor and attach nanobodies. And so this is sort of a quirk of evolution that alpacas, llamas, and camels have single peptide antibodies. And people have exploited this both for therapeutic uses, but also for basic science uses. So there's a company called Protein Tech, and you can buy these from them, and they have them tagged with GFP. And so you can label lamins, you can label actin, you can label vimentin. And so we thought, well, we could just take this nanobody and we put this sequence on each side. Now we have something which can bind onto a complex protein network and is no longer measuring tension on individual proteins, but instead is measuring deformation of those more complex protein networks. This idea actually came from my longtime collaborator, Teemu Ihalainen, who's in Finland. And he was the one who said, why don't you use these nanobodies to build this sensor? And this project was led by my PhD student, Brooke Danielson, who's since graduated, and Temu's postdoc, Bob. And it was a project that we did during COVID. And so I don't know, sort of has a fondness to me, right? As sort of the world was sort of burning during this time, right? We sort of had this continued interaction over Zoom to work on this project. And we thought the sensor could help us understand two questions, right? Are there forces on the nuclear lamins? And certainly there was a lot in the literature that really sort of indicated that nuclear lamins experience force. If you knock them out or perturb the nuclear lamins, a lot of things happen to cells. So I think it was sort of a foregone conclusion, but it was great to sort of come up with a tool where we could directly probe that. But I think the second question about where are these forces coming from and how are forces applied to the nucleus, right, was maybe a little bit, hopefully somewhere where we made an impact with this biosensor. So this work is published, so I'll just summarize it in just a couple slides. But this is, again, the idea that we had was the two nanobodies would create essentially a strain gauge or a spring where when it bound to the nuclear lamins, as that network is deformed, that would increase the distance between the fret pair and cause a corresponding drop in fret. And like Karsten mentioned during his talk, it's incredibly important to have good controls for these types of experiments. And so our major control in this paper is what we call a truncated mutant, or TM, control, where we only have one nanobody. And so hopefully in this case, the spring will never distend. And this gives us sort of an unloaded control so that we have an idea of how, what is the state of the cells with this sensor compared to ones where it's unloaded. So the first step was just expressing this. All this work was done mostly in epithelial cells, although we have some unpublished work that the sensor does work well in fibroblasts as well. And so you can see that our strain sensor is localizing really nicely to nuclear lamins. And so we confirmed that with a normal antibody for immunostaining for lamin AC. And then when we express this in MDCK cells, which you can see hopefully right away, is these pseudo-colored images for either fret efficiency, or here we're actually doing lifetime measurements. But you can hopefully appreciate they look different, right? And so that's really exciting for us right away, because one is telling us that the sensor is being stretched out, right? So it is operating as we sort of expected. And two, we have picked a protein that is mechanically loaded. And so just when we quantify this, you can see significant differences in the fret efficiency. So you see a drop, which again means that the spring is stretched out because this is an inverse relationship. So if you go in this paper, right, it's sort of painful to summarize such a long paper in just one or two slides. But we found a variety of things where the forces were actually different. And so we had to do a lot of controls also to sort of convince people that the sensor works. Some of the nuances about how the binding works and the affinity of each nanobody and the lifetimes of how long the binding is, right, sort of are a little bit ambiguous. Like we're actually surprised that this actually works. But we did some sort of, convincing experiments where we actually caused the nucleus to shrink by playing around with osmolarity. And this allows us to do paired comparisons of the same nucleus before and after. And then we did the opposite types of experiments where we actually compress cells using a gel and a cushion with a weight. And we're able to, again, look at the same nucleus before and after. And you can see in that case, the fret drops as the spring is stretched out. There were some obvious things, right, like external forces or actomyosin contractility that regulate the forces on the nuclear lamins. But there were some sort of unexpected things or some exciting things to us, which is that by playing around with chromatin condensation, we were actually able to show that the chromatin itself can regulate the forces on the lamins. So you can have both outside-in application of forces that affect the lamins, but you can also have inside out. And so one of the other things I just kind of want to highlight from that paper is some work that we did to show that, try to understand what were the spatial differences in forces in the nuclear lamins. And so one of the things that we struggled with is when we looked at our strain sensor, so that's the one that is dynamic and responds to forces, we were sort of confused that the strain sensor wasn't, sort of different at the periphery of the nucleus versus the center, or what I will call the nucleoplasm. Because you would sort of predict based on the staining that the lamins are more assembled at the edge or sort of periphery of the nucleus. And so you would predict that those would be the regions that would have the highest force. But instead, we sort of saw this homogeneous distribution of force. And at the time, we had made sort of a second sensor to try to show that this could be sort of a plug-and-play system, in which we swapped one of the nanobodies for lamin to make it a histone nanobody. And so now we have a scenario where it can bind lamins and it could bind chromatin. And we were sort of predicting that this would be bound at the periphery of the nucleus. But it turned out that this sensor was like accumulating a lot in this sort of nucleoplasm or sort of center space. And it also seemed to show a pretty good amount of tension in that space. And so when we read the field, and especially some of Roland Fossner's work, looking at the role of nucleoplasmic lamins, in particular lamin AC, there is evidence that these lamins, although they may not be that visible by immunostaining, are assembling and are important in sort of regulating the structure of chromatin and its mobility. And so we were able to use this sensor to better understand that those lamins that his group has been studying can also be experiencing mechanical forces. So this that's sort of the published work that we have, right, on this. And it really sort of made Teemu and I think about, could these nanobody-based sensors be used for other cytoskeletal proteins? And so one of the earliest ones that we made is we had the actin nanobody from this company. And we thought, well, why don't we make a force sensor for actin? And my student Bronson put this in fibroblast, and we were really excited because it labels stress fibers. And actin is under load. And about 5 minutes later, I realized like people will probably laugh at me, if I get all excited and said actin is under load, because this is sort of the basis for how sarcomeres work in muscle. And there have also been a couple other groups that have used similar approaches actually to measure forces on actin, where they have actin binding proteins that they put on each side of the TS mod, or the fret spring that I've been talking about. And I think, where this tool would be helpful is to maybe get an idea of spatial distributions of forces. If you have a fibroblast that's crawling around on the dish, if we could understand how the forces are different at the front versus the back, or how those forces change as the nucleus has moved rearward, that would be helpful. I don't think it's going to be helpful just to tell people actin experiences force. But where I think Bronson has really focused his efforts is really on vimentin, and trying to understand, is vimentin loaded and why would those forces be important? And so that's what I want to tell you a little bit about in the next part of this talk. So this is the same concept that I showed you earlier with the lamin force sensor. Now we have nanobodies that are binding to vimentin. And the idea would be, as the vimentin network is stretched, right, then we'll see a decrease in fret. We again have this control sensor that only has one nanobody, and hopefully right away you can see in our pseudo-colored fret images, right, they look different. If we go and quantify this, and this is lifetime measurements, so lifetime is sort of inverse to fret. So again, our TM will have the highest amount of fret or the lowest lifetime. And you can see our TS has reduced fret or increased lifetime. And so that, again, indicates that the spring is being stretched out. So it indicates the biosensor is working. And it's indicating that vimentin in a resting adherent fibroblast has some mechanical force on it. So we started to think about, okay, what could we do with this? And my group has always been really interested in the nucleus and the link complex. So we started to think about nuclear to intermediate filament connections. And if you know that field, you know that Nesbrin-3 is one of the key proteins, or perhaps the only protein, that is directly linking intermediate filament proteins like vimentin or keratin to the nucleus. But it's not really well understood why that would be important. So there was a group almost over 10 years ago, actually, that developed a Nesbrin-3 knockout mouse, and they're viable. And when you read the paper, there's not a lot that's happening in those mice. So that's sort of a disappointment. But then there was this really exciting paper that came out a decade ago from Ken Yamada's group, where they looked at how cells move in 3D. And of course, 3D is a very different environment, right, than a 2D experiment where cells crawling around a dish, and oftentimes it's very confining. And if you're not familiar with this story, I encourage you to check it out. But they talk about this concept of lobodopodial migration, and basically the nucleus, because the cell is so constrained, the nucleus almost acts as a piston. And so the pressures at the front versus the rear of the cell, the pressure at the front is much higher. And what was really exciting about this paper is Nesbrin-3 is required for this to happen, right? So you can think about maybe even the intermediate filaments ratcheting that nucleus back and forth. And then Alison Pattison, who has also spoken to this group, and I developed sort of a collaboration with her actually after she gave that talk, has some unpublished work really trying to get at this understanding of how vimentin is transmitting force onto the nucleus. So we thought, okay, why don't we start exploring this a little bit in our own group? And if we could just knock out Nesperin-3, right, we could create a scenario where now no longer vimentin would be connected to the nucleus. And we started some of this work actually before I moved to Ohio State. And I had a really great undergraduate, Jessica, who was helping us with this work. And she showed me some of the cells that when they had spread on fibronectin, this was maybe about 5 or 10% of the cells, had this really wonky phenotype, right? Where you can see this really large protrusion. And you sort of look at it, and because we don't have a movie, we don't really know is this cell, is this the front end or the rear end, right? So you don't know if the rear end is dragging or the front end is protruding and the nucleus is not able to move forward, right? But we thought that's really interesting. And so when Bronson started, he thought, okay, well, this would be better if we could sort of come up with sort of a fixed position where we knew where the leading edge and the trailing edge was. And so he got excited by these crossbow micropatterns. This is sort of well established in the field as sort of a model for how you can polarize the cell with front to rear polarity. And when he did this with our Nesmin-3 knockout lines, what he noticed is that the nucleus looks sort of wonky. I mean, it was sort of shifted. And it's sort of difficult, when you look at single images to actually understand what's going on. But the beauty of this pattern is it really allows him to aggregate the images, right? So he plots here the nuclear centroid positions, right? And you can see in the wild type cells, it's quite tight, right? They're sort of always in the same place, right? But you can start to really appreciate that there's quite a bit of deviation when you lose Nesperin-3. So a lot of our knowledge actually about nuclear positioning comes from the lab of Greg Gunderson. Greg had this really nice paper, many years ago, probably a lot of people are familiar with, where the nucleus actually moves rearward. So a lot of times we're taught that the centrosome is at the front of the cell when it's migrating. But the reason that happens is because the nucleus moves rearward, and that nuclear link complex, that actin to nuclear connection, is super important. And Greg had a follow-up paper actually where he did some interesting centrifugation experiments where he pushed the nucleus to one side or the other of the cell and watched it. And it turns out that the microtubules are important to bring the nucleus forward if it's too far backwards. So this was sort of inspiring to us, and we thought, well, has anyone really carefully looked at vimentin to nuclear connections and how those might be important? And so when Bronson did this, what he noticed is that it was really this side-to-side, or what we're calling horizontal displacement, and not this vertical displacement that was aberrant in these cells, right? So again, you can hopefully see it in sort of this cartoon, sort of summation of all of the centroids, right? That the side-to-side, you've got a lot of them, they're even off this crossbow pattern, right? And so that's what was significantly different, right? But the vertical or front to rear is not really significantly affected. So once we saw that, we thought, okay, that would be really cool. So let's go back and look at the vimentin forces. Maybe the vimentin forces are also going to be aberrant, right, if you don't have this connectivity. And so this is just a summation of what I had shown you earlier. Remember that our TS is going to be slightly higher lifetime, which means lower fret or tension. When we did this in the Nesperin-3 knockout cells, we got exactly the same results, right? To say that another way, there's no statistical difference between wild type cells and Nesperin-3 knockout cells with regards to the forces that we measure with our vimentin biosensor. So we've been thinking about this, what are contributing to the forces on vimentin? And so it seems like, vimentin is doing something to the nucleus, but it seems like the nucleus itself doesn't do something in the reverse direction to vimentin. And the other challenge that we've had with this project is while we consistently see large forces with our vimentin biosensor, we've had no luck finding ways to perturb that force. There's nothing we can do to the cells that actually reduces the force. So Bronson spent a lot of time working with myosin inhibitors, and that's traditionally what we would use with these biosensors, is inhibit myosin either using a blebistatin or a Y compound or ML7, and typically you would see sort of a relaxation of force, but we don't see that. And so we don't know if that's perhaps an interesting finding that once the vimentin network is set up and once the cell is spread, that even if you relax the actin, maybe these forces continue to persist for other reasons. And the other idea that we were just so sure would be a great control is we developed a plectin knockout line in the fibroblasts, and there's also no difference in the force on vimentin. Now, of course, there can be other linkers between actin and vimentin. So anyway, that's kind of where things stand. And so Bronson wants to look now at some more dynamic processes. For example, cells that are not attached or haven't spread versus cells that have spread, looking at the effects of substrate stiffness or even ECM composition. And just sort of a teaser, right, on sort of where we're going in the direction of keratin is my group has been most interested in epithelial cells. Keratin, of course, would be the intermediate filament of choice there. But we were sort of faced with this problem that there were no nanobodies for keratin. And so foolishly, not knowing anything about nanobody generation, I found a core facility at the University of Kentucky and spent a lot of time with them, and they sort of held my hand on what would be the process to immunize alpacas and develop and screen clones of nanobodies for reactivity to keratin. It's a little bit complicated, and so in the end, you have to have a very soluble peptide to immunize the alpaca, and of course, keratins are not soluble, so we did some experiments where we co-express K8 and 18, or we co-express domains of K5 and K14, and we have now what we think are nanobodies that react to either pair. We haven't yet screened them for cross-reactivity, so it's possible they just bind all keratins. But again, just as sort of a teaser, right, we've expressed this in a similar sensor with the two nanobodies. And you can see what looks to be quite good labeling of keratin networks in epithelial cells. And we believe we're starting to see some instances where we see differences in force, right? So again, this concept that the keratin network could be loaded, but when and where and how those forces are occurring is still a very open question. And this work is being led by a new student in my group, Sarah. So that's kind of hopefully the halfway point, right? And kind of gives you an overview of what we're trying to do in terms of building new tools so that we can understand what are the mechanical roles of intermediate filaments, in particular in living intact cells, right? There's a lot of elegant work people did a long time ago and still currently with looking at sort of the polymer physics of filaments as proteins like intermediate filaments and viscometer experiments and things like this. But I think what I really want to contribute is some tools where we can actually do this in living cells that can undergo dynamic processes. So for the second half of the talk, I want to really focus on desmosomes. And I think this is probably largely due to the influence of Andrew Kowalczak. So I stole this picture actually from his website. So hopefully he doesn't mind. I think it's a really nice picture that sort of highlights the sort of mechanical importance of intermediate filaments at the cell-cell adhesion. You can see that these filaments are very, very taut. So the idea that there could be mechanical forces on desmosomes and the keratin network in those regions is sort of compelling from that picture. This is another EM picture where, again, you can see how these structures are organized. If you're familiar with the field, you know it's similar to an adherens junction in that we have a transmembrane a cadherin, and then we have adapter proteins that link that cadherin to the cytoskeletal network. In this case, would be keratin. And then what you may or may not know is that if you have defects in desmosomal proteins and or keratin, it frequently leads to blistering diseases in the skin. And the explanation that's given is because these are very mechanically stressed tissues. So I think I already kind of went over the first point, right, which is that diseases that target desmosomes, typically we'll see those in the skin, but also I'll mention the heart. There are these sort of not so rare diseases where people run around playing soccer or run a marathon and they drop dead. Many times that is due to this arrhythmic right ventricular cardiomyopathy, which is where people have genetic problems in their desmosomal proteins. One of the questions, though, that I sort of struggled with is, I'm measuring forces on things like desmosomes or keratins, right? But why are these structures important and how can we help the field understand what they're doing? And because I study epithelial cells outside of the skin, I was really intrigued by the idea that your kidneys, the bladder, right, the liver, like all these sort of ductal and glandular structures that are made up of epithelial cells also have desmosomes. And I thought, well, why are they there and how are they important? And there's a lot of things that I'm summarizing in the field. This is other people's work about how desmosomes contribute to mechanical stability, about how you lose desmosomes as cancer progresses, about how desmosomes contribute to barrier function. They regulate the microvilli in the intestinal epithelium. And then there are people who are doing, I think, really good work to try to understand what is the mechanical relationship of the desmosome to the biochemical signaling. For example, looking at how changes in the desmosomes affect Rho GTPases or Notch signaling. And so years ago, when I had this first conversation with Andrew, when he was still at Emory, and he suggested I build a biosensor for desmosomal cadherin, I developed a biosensor for desmogliin 2. And this was sort of at the same time that Karsten, who spoke earlier, and Alex Dunn were developing their desmoplakin biosensor. And we both published around the same time. This work, I actually was able to do this work with a couple really talented undergrads in my group. And, sort of had a little bit of data that suggests that desmoglein 2 had a little bit of force, right? And in particular, the largest forces we actually saw were in cardiomyocytes when they were contracting. And Karsten and Alex's paper really suggested that desmoplakin doesn't really have force unless you stretch the cell, right? And at the time, I think some people were saying, well, you sort of sounds like you're publishing two different stories. They concluded one thing and I concluded another. And I really don't think that's the case. I think the forces we saw in resting cells were also quite small. And I think I agree completely with what Karsten has published and what he continued to say the other day about how desmosomes are not really loaded unless you do something to the cell, right? I also want to highlight some other work that I'm part of with Ruigo Yang, who's at Michigan State. He has an interest in this disease called pemphigus vulgaris, which is an autoimmune problem where the antibodies target desmoglein 3. That desmoglein is expressed mostly in skin. And it turns out that using some of the biosensors we had developed, that desmoglein 3 does also experience a low level of mechanical force in resting cells. And when these autoimmune antibodies are applied, we know that the barrier breaks down, the cell junctions break down. And his data suggests that that breakdown is actually due to a relaxation of contractility of the cell, in part by inhibition of Rho A. So back to this question, why are desmosomes needed in epithelial cells, in particular outside of the skin, right? And so when I think about this, I tend to think about cell adhesions more broadly, right? So we have adherence junctions, we have tight junctions. And in particular, we know that the adherence junctions are incredibly important in forming barrier function, in forming tissues, and in forming things like homeostasis of these structures in the body, but also dynamic processes like morphogenesis or branching. And so I sort of set out with this hypothesis that desmosomes, just like an adherence junction, also could play a role in these sort of fundamental epithelial biology processes. And so we set out to come out with approaches where we could deplete desmoplakin, which is one of the key linkers between the desmosomal cadherin and the keratin network, to prevent these connections. And I'll show you a little bit of in vitro data and maybe a little bit more in vivo data that we have where we're studying these processes. So just to kind of orient you, right, if you're not familiar with the desmosome, again, there's this transmembrane desmosomal cadherin. We have these linker molecules like plakoglobin, plakophilin, and desmoplakin is this large rod-like protein that makes the final connection between this structure and the intermediate filaments. Though a lot of work has come out about desmoplakin from Elaine Fuchs' group in terms of using mouse models. So a total knockout of desmoplakin, the mice is embryonic lethal. And then they went back to a conditional knockout in a follow-up paper where they knocked it out, I believe, using a K14 Cre promoter. And the mice were born, so there was no longer a developmental issue, but as soon as they were born, the skin fell off, and so therefore they didn't survive. Kathy Green has also done a lot of excellent work, also looking at the role of desmoplakin, and she has a dominant negative desmoplakin also to accomplish similar things. So in terms of the in vitro work, we decided to look at MDCK cysts or spheroids or acini, sort of depends on your background, what you call them. And so this is true for a lot of epithelial cell lines. You can grow them on tissue culture plastic or glass, and they look like nice monolayers. But if you put them in collagen or matrigel, they will form spontaneously these spherical structures. Sometimes I call them soccer balls, where you have cells on the outside. But in contrast to a soccer ball, this is going to be a water-filled lumen. And this is just a confocal section of the same spheroid, right, just to show you that it is a closed system. And a lot of people, including myself, would argue these would be good models for ductal or glandular structures in vivo. And so before I get to the desmoplakin part, I just want to highlight some work that we had that might help explain why we thought this was an important situation to look at desmosomes. So many years ago, I had a really amazing student, Vani, who decided to use our E-cadherin biosensor. And it's not ours. It was originally developed by Alex Dunn, but we had gotten it and we had made a stable cell line. So she said, why don't we use this to study the differences in force at the junctions between 2D and 3D. And sort of another sort of unexpected result was that the mechanical forces were higher in 3D. And the reason this was sort of unexpected to us is because when you grow them in matrigel or collagen, these are very compliant networks, right? So the ECM is much more compliant or less stiff. And so we thought, how can cells in 3D generate more force in a more compliant ECM? And it turned out that this was due to osmotic gradients, right? So epithelial cells undergo directed transport of ions. This is the basis, for example, how the kidney filters. But it turns out all epithelial cells like to pump ions apically, in particular chloride ions. And so this was creating a situation, much like the soccer ball metaphor I mentioned earlier, where this lumen space becomes pressurized, right? And that pressurized lumen allows the cells the ability or provides resistance against their own sort of internal contractility. So we thought, okay, this could be a really interesting way to study desmosomes and their importance in sort of a mechanically challenging environment, right? And so we had, again, another undergrad many years ago had helped me develop the cell line in which we depleted desmoplakin from MDCK cells, and then we grew them in matrigel. And you can see that we've lost the desmoplakin staining, and potentially maybe we had some changes in morphology. And so Sarah, who I again mentioned recently had joined my group, has been working with these cells again and trying to understand, are there differences in these cells? And this is some recent data that she showed me where we're sort of seeing what looks like accumulation of something inside the lumen where it looks a little bit different to us, but we're not really sure what it is. And it's not reacting with the DAPI, so it's not, it could be some cell debris, right? But it's not cells itself, right? Because we don't see anything. And so Sarah's going to do some experiments where we play around with pressurization of these structures to try to understand if loss of desmosomes is important in sort of the mechanical stability of these 3D spheroids. And then the second part about the desmosomes that I want to talk about is how we could try to understand how these structures could be important in branching or patterning. And Andrew Kowalczyk had pointed me to some really excellent work that Al Reynolds had done. And if you're not familiar with Al Reynolds, he passed away a couple of years ago, but he was sort of the person who led the field with regards to this P120 catenin, which is really important in sort of the stability of the adherens junction. And he had these two excellent papers where he depleted P120 catenin in a conditional knockout mouse using the MMTV Cree. And this is a Cree that's highly expressed in the mammary epithelium and also the salivary gland. These are two structures that undergo branching morphogenesis. And so I thought, okay, I'm an engineer. We haven't done a lot of mouse models, but I could maybe just reproduce what he had done with P120 catenin, but looking at desmoplakin. And the hope would be we would see also some disruption in those structures. So we set out to develop a desmoplakin flux model and cross it to an MMTV Cree, again, with the idea that we could understand what are the role of desmosomes in mammary epithelium and the salivary gland. My postdoc Pepe Wang has been leading this project, and we've been getting some help from our veterinary pathologist, Kara Korps, here at Ohio State. Fortunately for us, these mice are born at Mendelian ratios. We do see some loss of the pups early, sometimes in our conditional knockout. What surprised us, though, initially in this study was that the mice were frequently smaller than the control litter mates. And so I got all excited. I started thinking about, the pituitary gland and are there desmosomes in the thyroid and all these things, that could lead to maybe a dwarf mouse. So we sent them to Kara for a necropsy. And she, once she got to them, she called me up and we had a Zoom because she was quite concerned because she said, your mice are infected. And you got to report this right away to the animal facility. And I was like, what's happening here, right? But it was only the knockout mice. The control mice were perfectly fine, even though they had been in the same cage. So they have these dry, crusty skin lesions that are indicative of a bacterial infection. We also have elevated neutrophil counts in these mice, right? So they do seem to have some sort of systemic infection. And Pepe has sent so far just one, right? So it's preliminary, but we did this microbiome analysis with 16S ribosomal sequencing. And so in our knockout mice, we got 90% of the hits were Staphylococcus, whereas in our control mice, you just get a whole wide range of a microbiome. That sort of makes sense because we know that staph bacteria are known to target desmosomes. So certain strains of staph can secrete these exfoliative toxins to break down the desmosome and allow the bacteria to go deeper into the skin. If we put the mice on antibiotics, things improve. If we trim the nails, that also seems to help with skin irritation. But of course, we're sort of balancing sort of the ethical concerns as well, and sort of the challenges also of working with mice that seem to harbor skin infections, creates some friction between us and sort of the veterinary staff who are helping us with this project. But, the real question was, I just told you we were trying to build something that didn't target the skin, right? Because we don't want skin issues, and I want to understand things outside of the skin. So why on earth is there what we think is a skin phenotype? And so I apologize if maybe this is too graphic for a few people, but I just want to highlight sort of the severity of these skin issues. So there's a lot of issues around the ear. You can see some of the sort of issues with the skin. You can also see sort of this dry flakiness. We don't know if it's sort of a dandruff or perhaps dried sebum, which is the oil that's secreted in the skin. And if we remove the hair, you can sort of, again, see these sort of like skin lesions, right, that we think are bacterial in nature. So Pepe took our MMTV Cree and crossed it to a TD tomato. And so this is a situation where the Cree will drive excision of a lock-stop-locks site in front of the tomato and will turn on. So any tissue that is red, right, at some point either had or has Cree activity. And when we looked at the skin, we see some Cree activity at the very outer surface of the skin, and presumably some of the keratinocytes. But what was interesting to us is we saw really large amounts of Cree activity in what are the hair follicles and the sebaceous glands in the skin. Now, when we do an H&E staining of the skin, if you sort of focus your attention here, this is the top row. So we have dorsal and ventral skin, and the top row is the control animals. You can see, again, these hair follicles, and then the more lighter cells, I'm told, are the sebaceous glands right here. You can see, again, sort of, regular spacing of these. But when we looked at our knockout mouse, what we saw are much, much larger sebaceous glands, increased numbers of sebaceous glands, and scenarios where now our sebaceous glands seem to be on sort of the wrong side or sort of random positioning around that hair follicle. So really a quite a dramatic dysregulation in sort of the development and or maybe perhaps maintenance of sebaceous glands in the skin. This is just another sort of quick slide about the salivary gland. It looks different to us, but then one of the challenges I've sort of faced as we sort of try to dabble in things that we don't fully understand is, yes, the salivary gland looks different, but I'm having a hard time understanding or being able to articulate what that difference is and why it's happening from loss of desmosomes. I'll share with you just maybe two more little snippets about the desmosomes as I sort of wrap up today's talk. We also thought that we should look at desmosomes in the kidney. And I thought this will be a really simple experiment because the kidney is not important in embryological development, right? So if there was a defect, the pups would be born, and then we could do some analysis of the blood chemistry and try to understand if kidney function was impaired. It turns out that using a different Cree, so we use KSP 1.3, which deletes, which expresses in some of the proximal tubules in the kidney, the mice appeared normal, and we sent these kidneys to the pathologist, and she told us there's absolutely no difference when you lose desmoplakin in the kidney. And we thought, this is really crazy. How is this even possible? When I dove into the literature, there was another group that has done, I guess, sort of similar approaches where they don't delete desmoplakin, but they're deleting a desmosomal cadherin, desmoglein 2, also has minimal effects on kidney development as well. So then we thought, well, why don't we challenge these mice kidneys with an obstruction model? So one of the ureters is obstructed, it's sort of a mimic of a kidney obstruction. And you can see that that obstruction causes, sort of severe injury to the kidney. And again, similar sort of setup here. The control images are on the top. So the first one here, the right kidney is not obstructed. You can see it looks quite normal, and you can see in the knockout very minimal differences, if any, between the two. But then in the obstructed kidney, which you can see is in the control mouse, you start to see, again, some damage to the kidney, some spaces where we lose cells, right? But when desmosomes are deleted in this other mouse, you can see we get much, much larger sort of dilations and almost like a Swiss cheese sort of look to the kidney. So what this indicates to us is that the lack of desmosomes creates a scenario where now the kidney cannot respond to a sort of dramatic overpressurization in an injury model. the last place that we kind of want to go with the desmosomes is I happen to meet Jerry Apodaca, who's at the University of Pittsburgh, and does really elegant work looking at the sort of cellular biology and mechanics of the bladder. And before I had met him, I'd never really thought about the bladder as a mechanosensitive tissue, right? But it undergoes really, really large strains between filling and voiding. And Jerry had this really nice paper that came out this year. I just want to kind of highlight for the field, where he looked at the keratin networks in the umbrella cells. So these are the cells that form the bladder, and looked at them in filled versus unfilled bladders. And hopefully what you can kind of appreciate is in the filled bladder, you have really nicely taut keratin filaments, right? So again, could there be a role for desmosomes and/or desmosomes to keratin filaments, that anchorage of the keratins at cell-cell adhesions in the bladder? And so I hope in another couple of months, we'll have some data here to show you, are desmosomes important in the bladder? So just to wrap up with some final thoughts, hopefully, between my talk and the really excellent talk of Karsten, you have a sense of that FRET force biosensors are really excellent tools to study things that might have force, including things that are either the intermediate filament network itself or things like desmosomes that are connected to it. And hopefully my data is enough to convince you that vimentin and keratin and lamins experience force, but that we're still struggling, both me and I think the entire field, of why those forces are important and when and where those forces are happening. And then the second half of the talk really was to highlight that desmosomes, which we all know, if you're sort of in the know about cell-cell adhesions, are really critical structures in things like the skin and the heart. But how those structures could be important in other tissues is not well understood. So it was real surprising to us that kidneys would develop normally without a desmosome, but probably wasn't so surprising to us that in an injury model, the lack of a desmosome would be a detriment. And then maybe what would be surprising to all of us, right, is that desmosomes seem to have a really important role in sebaceous gland development and maintenance in the skin. And I think that's something that we will certainly want to follow up on. So with that, I hope I highlighted all the important people that were involved in this, my students who've done a lot of the work, and then in particular, some of the collaborators we've had on this project. We've had other collaborators on other projects, but I don't have room to acknowledge all of them. And of course, my funding that comes from NIH, I'm funded by General Medicine, and my really excellent group, both when I was at Virginia Commonwealth University and now that I'm at Ohio State.